Hello, my name is Alena Brown. I work with Delaware EPSCoR's Project WiCCED on the distribution of cyanobacteria in Delaware ponds. So to start off, cyanobacteria are photosynthetic microorganisms that are autotrophic and can fix carbon dioxide for their growth. They're known to produce some of the largest blooms in lakes and ponds, and they can also negatively affect the water quality around them. Also, they are known to poison livestock and humans as well. So my project aims to examine cyanobacteria that blooms in Delaware ponds to potentially determine if they are toxic or not. And through this, we've developed some methods of analysis through cultivation-dependent methods and cultivation-independent methods. So some information on my background here. The samples were collected at the Coursey Pond Dam site. And here I have a picture of the pre-bloom at the start of my project and the actual bloom as we moved further in towards my project. As the weather got warmer, you can see that the bloom became more abundant in its pigment. So we want to ask some overarching questions. What types of cyanobacteria are present? And through PCR sequencing, do the isolates carry any toxin genes? And then how many bacteria can we grow? Can we isolate important bacteria for future studies? And lastly, what toxin genes actually exist in Coursey Pond? So for my methods of study, like I said, we developed two methods of analysis, cultivation dependent and cultivation independent. So for my cultivation dependent, we had samples from Coursey Pond collected, and we wanted to run a dilution to extinction series, where we have inoculum plus any growth media, which was nitrogen, phosphorus, trace elements, and any vitamins. And this analysis will allow me to observe through sequencing any of the positive PCR products that we obtain. For the cultivation independent method, we ran those same samples that were collected from Coursey, and we wanted to extract the DNA from the preserved samples and then quantify that DNA to eventually examine those toxin genes and the 16S gene as well. So with this, we can say if the PCR is positive, then there is potential for the toxin genes to be present. And we can ask what types of cyanobacteria are present, and do these cyanobacteria carry any toxin genes? So we've also acquired Coursey Pond environmental data from my project, given by Scott Andres of the Delaware Geological Survey. Each of the arrows here indicate when the samples were collected for my project, and we collected data on chlorophyll A, dissolved oxygen saturation or percent saturation, and phycocyanin. We also collected data on temperature, which was important to look at because cyanobacteria are actually most abundant when in warmer temperatures. And we also collected data on nitrate as well. So when looking at my environmental data here, we noticed that the phycocyanin reached its highest peak on about 8.6, which is pretty interesting because if you look back at my background here, the bloom had most pigmentation at around 8.6. And so this is because phycocyanin plays an important role in the producing the pigment of cyanobacteria because that's what gives it its blue-green color. So cultivation of microbes from Coursey Pond. With this, we saw that later samples appeared to have the higher number of cultivatable microbes, and that's because of the added nutrients, the nitrogen, phosphorus, and other trace elements. And we saw the earlier samples did not recover those more viable microbes than those with the added nutrients. And so moving on, we ran transfer of those pigmented strains from Coursey Pond. And so this is an example here of our 9/11 sampling date. And we ran these strains from primary, secondary, tertiary. And then there was a split into synthetic freshwater media and Coursey Pond H2O. And with this, you can see that the further we got down into the tertiary strain, you can see more pigmentation. And so in microbiology, this is a subculturing technique that is used usually to grow and maintain bacterial cultures to go from a mixed culture to a more pure culture. And that's what we wanted to do here. So if the bacterial cultures are left in the media too long, they will take up the viable nutrients. So we wanted to ensure that unwanted bacteria is kept out of the important culture. And so this technique allows us to see if we can maintain these cultures in the lab for future research on what may trigger toxin production. And with this, there are also represented cultures for the date 820 as well. So primers for toxins potentially present in Corsi pond. So with this, these primers were found through literature, and we want to amplify toxin genes through PCR. And hopefully with this, none of the samples or isolates will turn out positive because we want to observe something that is unique to Delaware. And the intention with these primers is to be able to say whether or not these toxin genes will be present in the community or the cultivated strains. I also developed database sequences for toxin genes in Corsi pond. So with this, I am building a database sequence for each of the toxin types and for cyanobacteria specific genes to say whether or not any sequences we recover are unique to Delaware or have been observed before. So for example, in figure 6, you can see here I have a gene for slender spermopsis. So for example, we want to observe something that is unique to Delaware. So say if we obtain something in the lab similar to slender spermopsis, and we found a sequence for slender spermopsis through literature, and that can indicate that this slender spermopsis gene could be potentially present in Corsi pond. That is good, that is positive. However, we want to find a toxin gene that is unique to Delaware. So we want to find something that has never been observed before in literature, that has never been found. So that way we can move forward with future research. Figure 7 here is a database that shows the number of sequences that I have for each toxin type. And for example, I have over 100 sequences for saxitoxin, or I have 47 sequences for slender spermopsis. And what I want to do with this information is eventually I want to create a phylogenetic tree. And I want to ask, what does this potentially tell us about their function? And this is because each gene here has its own separate function. They have their own different evolutionary trait. And so using this, the toxin genes collected will be used for database sequencing and CLC main workbench. In conclusion, we know that we can cultivate cyanobacteria strains. These strains can be transferred to synthetic freshwater media. I have identified useful primer sets for toxin genes and cyanobacterial marker genes for PCR. I have constructed databases to analyze any sequences from PCR to see if the strains are unique to Delaware or related to any other blooms that are in the region. And so this brings me to my future research. With this information, now I have the tools to address the questions that were asked in the introduction once we can get back to the lab. So I will continue cultivation to isolate model cyanobacteria for laboratory experiments to identify the conditions that will induce toxin production. Also, I'll continue PCR and sequencing to identify what environmental conditions are associated with toxin gene presence in Corsi pond. If you have any questions, please contact me via email. And I would also like to thank Scott Andres and the Delaware Geological Survey for providing me with the environmental data for my project. Thank you to Alexa Bennett and all of the lab members of Hanson Lab for the comments on my poster drafts. And finally, I would like to acknowledge Dr. Thomas Hanson for your help and guidance through my time working on this project. Thank you very much.